So now we come to the third part of this lecture. Going to be not so long. After the first two lectures, everything has been discussed, which was basic and a little bit advanced. In this image, we saw the stem cells in the crypt, which were repairing and rather replacing. So the repair was to replace the old cells, as the half-life is 4 to 5 days, and of these small intestinal cells, and this was keep jumping up after when it was followed in a pulse chase assay. So with the pulse, you could see they just give this dye, the stain, and then they were just chasing these cells after time 0, 1, 2, 3. And eventually, these cells were moving up. It was telling that there are new cells being produced, and that they were replaced as these cells are worn out, they turn over. Identification and isolation of stem cells is not easy. It's a very difficult job to do. How do you isolate a stem cell, and actually how does it look like? So how a stem cell look like? There are very isolation of these stem cells is very difficult. The reason is actually they are very rare, and since they are very rare, they are very low in number, or the other way around. They are low in number, that's why they are very rare. For example, you could see that the percentage of hematopoietic stem cells, which we always commonly refer to as one of the classical examples of stem cells, is 0.001%. So not one cell in 100 cells, but not one in 1,000, not one in 10,000. One in 10,000 cells is actually hematopoietic stem cells. And the reason why we cannot really make full use of these stem cells in tissue and organ engineering or in other therapeutics lies in the fact that they are really difficult to isolate, and that is totally reliant on the fact that they're very rare and low in number. Nonetheless, in isolation assay, we can do one of the method to do is through FACS, fluorescence associated cell sorting. So that's a flow cytometry technique in which we do sorting cell by cell. So we used the cell surface receptors, and we make these tags, protein tags, which are also known as antibiotic. Like you see this is a protein tag, and these are antibiotic, and they have a dye, and they go and bind to the cell surface receptors of cells, and then we sort these cells individually. So what happens in this flow cytometry FACS is that each cell would be pushed down this tube, one cell. So the diameter of the tube is adjusted in such a way that only one cell can actually come at a time. And when this one cell is going to these positive and negative electrodes, there will be a charge on them because of the dye. And because of the dye, there will be it will absorb a different wavelength of laser or light. And in these cases, there will be differences in respect to charges, and with respect to charges and with respect to absorbance of different wavelength of lights. And then there will be, in this case, at least two negative and positive samples would be set, and then they will be separated. And then you can analyze these single cells for stem cell properties and determine if there are stem cells or not. Another one of the other method is to identify stem cells is you label the living cells with molecular markers as discussed here. So this is a molecular marker. And then determine the specialized cell types they generate. So what do you do then? How do you do this? You remove those particular cells from an animal, and then you label them in cell culture. But what happens next is like you transplant them back into another animal to determine whether the cells replace, I mean, if they repopulate their tissue of origin or not, because that particular tissue of origin, which we say, if this is like blood cells, so this second animal, this second animal would have no capacity to make blood cells. So a bone marrow transplant is one way to isolate and identify cells by exposing animals to radiation so that their bone marrows are no more working and they're no more making these blood cells. So that when you have a bone marrow transplant, you have hematopoietic stem cells within the bone marrow, which start replacing and you start repopulating. And this is how you can do it. One of the methods. There are more methods that are very sophisticated, and I think it would not be wise to discuss and spend so much time on those sophisticated methods for your levels at the moment. So the last part of our lecture is the applications of stem cells. There are many of applications of stem cells, organ and tissue regeneration, as we already discussed, in case of burn victims. So there's a tissue regeneration which causes skin. Similarly, you can actually regenerate organs. There are some organs transplants going on around the world. For example, one of the common and everyone knows about it is the kidneys transplant, but there are not enough donors. Plus, then you have to do a lot of matching and cross matching, and if the mismatch happens, people can die because of clogging. But this is a general figure diagram for the application of stem cells. A lot of stem cells are used in research, cell therapies, target drug validation. I don't know how do they do it, but then 3D printing, yes, this is and drug delivery, toxicology screening, a number of not only therapies, but at the moment in our applications, we are more focusing on therapies. Similarly, theoretically, you can use stem cells for getting rid of Parkinson and Alzheimer's diseases because the brain tissues that are damaged using stem cells, you can actually regenerate them. And these treatments are in the lab these days. A number of scientists are researching on them. Not really in the market to be performed, but this is a potential. Healthy heart cells, actually it has been developed, and again the work is in its infancy, but this is possible that you have you repair the damaged hearts, so it will actually repopulate the heart with the healthy tissues. And similarly with the in case of diabetic patients, you can have pancreas, so you can repair pancreas, and if you repair pancreas, with the insulin properties will be perfectly fine, and they would need no insulin injections. So, but these are all the applications that we might see, or might our children, or maybe your children see, because this will still take a couple of decades to come. But theoretically, this is all very possible. Similarly, in cardiovascular diseases, it's the same story, and I have taken the paper of NAS in early July 2013 when they created blood vessels using mice in lab. Similarly, these blood vessels are very important for cardiovascular diseases. So if we can create these kind of cardiovascular blood vessels, they would be replaced with the one that are clogged, due to which you have a lot of cardiac diseases, a lot of heart failures and deaths. But again, this is just an infancy. So hope this will be one day a reality. That was all for today's lecture, and I would now I have something more. Just give me a second.